Genbank considered to be as an example for nucleotide sequence databases. So actually this Genbank is maintained by NCBI. So you can type NCBI and Genbank. So if all of you having a computer, you can also follow me by typing these particular keywords so that you can follow and you can see whether it's working or not. Okay. So NCBI, as you all know, that it's a National Institute of Health maintaining this NCBI, that is National Center for Biotechnology Information. That is the database. It is maintained by National Institute of Health. It is in the National Library of Medicine. That is what the website address. You can see the website address: NCBI, NLM, NIH, Genbank. So it means that the NCBI is the database. It is present in the National Library of Medicine. It is maintained by National Institute of Health. It's a government organization. So that is how this particular Genbank is located. So even though the name says Genbank, you can see here it's a nucleotide because Genbank is a nucleotide sequence database. Clear? So you can see here Genbank, but here you can see a nucleotide because the Genbank contains nucleotide sequence. Similarly, the nucleotide sequence is available in DNA form. Okay. In this particular National Library of Medicine, you can see many other databases. You can see here, these are the other databases which are available in this particular NCBI. We are going to see only one database now, but if you are interested, you can see all these databases. Okay. So here, for example, you can see PubChem is going to be here only. PubMed is going to be here only. You can see here PubChem and PubMed. So these are the other small nuclear chemical database is a PubChem and literature database is a PubMed. Here also structure database is here. And you can also, if you want a protein sequence, you can check this particular protein sequence databases. For journal article, you can check this PMC. For human genes, you can check this ARMIM. Okay, so this ARMIM is going to contains chromosome-based gene locations. ARMIM, Online Mendelian Inheritance of Man. It is considered to be as a secondary database. So even though we are going to see only one database now, if time permits, you can check all these databases and you can get information about different databases from this National Library of Medicine, that is from the National Center for Biotechnology Information Database. Okay. So now if you want to get any sequence, gene sequence, if you want a particularly full gene sequence, you can select, there is a gene option. If you want to know how many number of sequences related to a particular protein is available, you can give nucleotide. Okay. So now if anyone suggests any particular protein, I will type that name. Please give one suggestion from yourself. Students can give. Any one protein name.
CRP.
CRP.
Yeah, C-reactive protein.
Okay. See, it will list. Okay. So when you are seeing this list, you can see the word here, complete CDS. Okay. You can also go through different parts. You may not, in some cases, you may not have this particular word, complete CDS. Complete CDS means complete coding sequence. Okay. So that link alone give a complete nucleotide sequence. Otherwise, you will get partial CDs also. If you go to some other page, you can see that you will get partial CDs. Incomplete gene also will be there. But for our purpose, it will all shows a complete CDS. So you have to check that. And one more thing is that we can also decide from which organism we are going to select this particular CRP from here, species. So if you want to get sequence from particular microbe or particular organism, you can select from here. You can also see here also some of the organisms are listed. So you can select this particular gene from any particular organisms. See, if you want to get this particular protein from human, you can also type Homo sapiens along with this particular sequence, or here you can select through this particular species link. Okay, so if you want to need humans or Homo sapiens, you can add that, you can get it. So now, so like this, we can filter this particular searches. So now you can see Homo sapiens. That also it is a complete series there. Okay. So like this, we have to find out the particular nucleotide sequence with particular organisms. Clear? Then we can select any one of the sequence by reading this particular topic. Okay, you can see, go through this. There are several. Even variant is also there, transcript variant of this particular gene. You can see this variant 3, variant 1, variant 2, and complete gene is also there. So you can check all these things and then select our interest gene. Okay. See, this is what actual nucleotide sequence page of this particular CRP gene. So CRP gene, it's a complete CDS. It is also considered to be as a pseudo gene. See, this is the original sequence of this particular CRP. So when you go through this particular sequence, you have to note this particular number because this is considered to be as an ID. It is considered to be as an ID for this particular sequence. If you know the ID, it is, since the database is Genbank, it is called as a Genbank ID. Okay, so here you can see 10 AF 44 28 18.1. Okay, so that means what? It's a first version of this particular sequence. So if the author changes some of the sequences of this particular sequence, then this version will change. If they modify any of this content, they will change this version as 0.2 so that we know that something is changed. So for that purpose only this version is indicated here, and this is considered to be as a Genbank ID, and this is point indicates the type of version of this particular sequence. And then this is the Genbank format of this particular sequence, and here all the Genbank file starts with the word locus and it will give information, annotations about the gene. You can see that the source of this particular sequence and organism of this particular sequence, taxonomy classification of this particular sequence, and then who identified this particular sequence, who submitted this particular sequence to the databases. They are all given as a references, and publications related with this particular sequence will be given here. Then the translation, features about this particular nucleotide sequence. If this nucleotide sequence is translated into protein, then what is the type of protein you will get and what is the sequence of this protein is also be listed here. And then ID, that particular protein ID is also listed here. And then the word comes origin. Okay, so after the word origin, the sequence, original sequence of the CRP started. Then in each line, you can see that 6 blocks, 1, 2, 3, 4, 5, 6. So 6 blocks, each block will contain 10 nucleotide. So one line will contain 60 nucleotide. Okay. So like that, the sequences are listed here. And then at the end of this particular sequence, you can see a double slash. So the double slash indicates that the sequence completed, ended. The end of this particular file is indicated by this double slash. So starting of the sequence is shown by the word origin. So this is the actual sequence part. Okay, so before this, all these things are called as annotations, information about the sequences. So this is what the nucleotide sequence available in Genbank. Say if you want to convert this particular sequence into another format like FASTA, you can click here as a FASTA link. It's another format. So this is called as Genbank format and this is called as a FASTA format. So if you click here, it will convert this particular sequence in the FASTA format. Okay, so if you click here, see now the sequence is converted into a FASTA format. FASTA format is nothing but greater than symbol followed by one line description about the sequence. See the annotations in the previous page we saw that more than half of the page annotations is there. Now the whole annotations is converted as a single line here. Then the sequence also present continuously after the first line of this particular greater than symbol. So greater than symbol, one line description about the sequence, and from the second line you can see the sequence. So this is known as the FASTA format. What is the speciality of this FASTA format? If you want to analyze, if you want to submit the nucleotide sequence to any tool, bioinformatic tool, so those tools are like this particular sequence format. So for analyzing sequences, so this FASTA format is more favorable. If you want to know about the sequences, then the Genbank format is more useful. Okay, so that is what the difference between these two formats. And then if you want to save this file, so we usually save the file by using normal saving options, but here you have to use this particular link. You can see the send to option here. You have to select send to option and you can select complete record and then you have to select file and then here you can select the format. So you can download any one of this format. You can see that Genbank format is here, FASTA format here, ASN format here, XML format here. So you can select any one of this format to download. Mostly we will use Genbank and FASTA. The ASN, XML, these things are all used by the computer science people for building up databases and other things. They will use these two formats. For bioinformatics part and biological studies, we will use both this Genbank and FASTA format. So first we will select Genbank and then create file. Now the file will be downloaded. You can see the file is downloaded. So you can see the end of the file. The file will have sequence.gb. GB means Genbank. So the file is saved in the Genbank format. So that is what it shows. Like a Word file will have an extension of DOC, doc or docx. This particular file will have a GB, Genbank extension. So if you want to see the file, you can right click and you can open either through Notepad or you can use a WordPad to see the content of the file. Okay, see now. So this is the file we have downloaded from the nucleotide sequence. So this is in the Genbank format. Okay, like this you can also download the sequence in the FASTA format. Okay. So this is how we can download or retrieve sequence from the Genbank. Clear?